Good day, folks. Welcome to my channel. I'll be discussing about the natural cause and the immunology of AIDS in this video. The natural cause of the disease consists of three stages. The first one being the window period, the second a seropositive stage, and there's a third stage. Now let's see all these stages one by one. The first stage is a window period. So here, when the virus enters the body, it is multiplied in the body cells, but it cannot be detected easily. This is called the window period. The viral capsid antigen P24 can be detected in blood during this time. So here the P24 antigen assay yields you a positive result, whereas all the other tests such as ELISA gives you a negative result. The second stage is the seropositive stage. So here the main thing what you have to note here is the antibodies are seen in circulation. So thus it is known as seropositivity. So the antibodies of the HIV virus are seen in the serum. And this takes place after a few months. And during this period, the person is completely normal. However, this person is a carrier of the disease and can transmit the disease to others. So basically, the window period it can last from 2 weeks to about 2 months, and the seropositive stage takes place after this. The third stage is when the clinical manifestations start. So in the second stage, the person is a carrier, but there is no clinical manifestation. And in the third stage, it is the stage where the clinical manifestations start. And by this time, the immune status of the individual is so depressed. So all the other opportunistic infections are affected to the patient, and the patient usually succumbs to death within about 2 years after entering this stage. So this is a graphical representation of the three stages and also representing the concentration of the antibody in the blood, also the virus capsid antigen. So you can see from this graph that in the first few months, that is about 1 to 5 months, that is during the window period, the concentration of the virus capsid antigen is high in the serum, whereas antibody levels are low. And once the antibody levels are created after about 5 months or a year, this level of the antibody rises, and after about 7 years it comes down and the antigen is again increasing. So this represents the decremental immunity status in the body. Now we'll look at the immunology of AIDS. So here the CD4 T helper lymphocytes are decreased in number. So we have already mentioned in the previous video that the CD4 receptors which are present on the membrane of the T helper lymphocytes act as the major site where this HIV viruses bind. And this binding takes place with the help of the glycoprotein 120 which is present on the virus particle. So this GP120 surface unit could specifically attach by CD4 molecules present on the surface of T helper cells, and therefore HIV preferentially enters into the T helper cells and they are lysed. Now the macrophages and monocytes act as reservoir of HIV infection. Though it is said that initially the HIV is affecting the T helper cells, there is a difference to this fact. Now let's discuss about the immunoselection of virus. So in the early phase, the HIV it enters the macrophages, not directly to the T helper cells. So the HIV enters macrophages through a receptor which is jointly made by CD4 and CCR5 proteins on the surface of macrophages. So initially that is how the first binding takes place. And the HIV surface antigen, which is a GP120, it has a perfect fit only for this type of receptors on the macrophage. It is not initially binding to the T helper cells. And this type of HIV is known as the M-tropic HIV. So in the early period, lymphocytes are spared. Now after a few years, the GP120 is mutated. So that dual trophic viruses are produced, and in the late phases of the disease, T-tropic HIV is generated. So this T-tropic HIV is the HIV which have the mutated GP120 which can specifically bind to the CD4 receptors which are present on the T helper cells. And these T helper receptors, they are made up of the CD4 and CXCR4 proteins. So the T helper lymphocytes are preferentially killed, leading to the disease manifestation. But as it is initially entering the macrophages can act as a reservoir of the HIV infection. So now to talk about the genetic resistance and the genetic heterogeneity of the virus. The genetic resistance is in about 1% of the population. They are resistant to the HIV infection, and this is because there is a mutation in the CCR5 protein receptor which is present on the surface of the macrophage, to which the HIV initially binds. So when an individual has a mutated CCR5 gene, that person escapes from the HIV infection. Now, the genetic heterogeneity of virus. There are two types of HIV, HIV 1 and 2. The most common one is the HIV 1, which is a predominant one, and each type exhibits remarkable microheterogeneity, and the basic reason for the difference in antigenicity of the virus, or why a drug could not be developed for completely curing the HIV infection, is because about 15% of amino acids in the envelope, which is of the GP120, they are highly variable. So there will be mutations each time, and a particular drug cannot be used to resolve the HIV infection. That's it for this video. If you honestly did understand the topic, do like, share and subscribe. Thank you.